Hello again, and welcome to the Honors College Fall Undergraduate Research Showcase. I'm Toni Doolen, the Dean of the Honors College, and I'm very excited to have so many members of our community who've joined us tonight for what I'm sure is going to be an absolutely terrific set of presentations. Before we start the program, I'd like to acknowledge that Oregon State University in Corvallis, Oregon is located within the traditional homelands of the Marys River or the Ampinifu Band of the Kalapuya. Following the Willamette Valley Treaty of 1855, Kalapuya people were forcibly removed to the reservations in Western Oregon. Today, living descendants of these people are part of the Confederated Tribes of the Grand Ronde Community of Oregon and the Confederated Tribes of the Siletz Indians. You may have noticed that we're recording the presentation today. The final video will be edited to remove any identifying information about those of you attending as guests before anything is shared or posted. No aspect of the Honors College student experience is more impactful than research. Working with world-class faculty on research at the frontiers of knowledge is, for many students, the defining element of their undergraduate experience in the Honors College, providing skills, growth opportunities, and connections that resound for years after graduation. One of our great strengths as a college is that we can draw on the outstanding research and faculty across Oregon State University. Oregon State University received more external research funding than any university in Oregon. Last year, faculty were engaged in $383.9 million in grant-funded research, the fifth consecutive year that OSU research has exceeded $380 million. And we in the Honors College are fortunate that these research leaders are such willing and supportive partners, offering honor students opportunities and experiences unsurpassed at any university in the world. I'm so excited that one of these amazing partners has joined us to introduce our event by showcasing the work being done in one of the university's outstanding research centers, the Linus Pauling Institute. Dr. Emily Ho has been director of the Institute since 2020, and she is also a professor in the College of Public Health and Human Sciences, with research focusing on understanding the mechanisms by which nutrient status in healthy foods affect the initiation and/or progression of chronic diseases such as cancer. Prior to becoming director, she was the endowed director of the Moore Family Center for Whole Grain Foods, Nutrition and Prevention. And throughout her research and leadership at OSU, she has consistently driven research projects and collaborations that cross disciplines and communities, and she engages with both graduate and undergraduate students. In short, she is a model and, fortunately, also for us, a frequent mentor for honor students. Dr. Ho, thank you for joining us.
Thank you so much for having me. As Dr. Doolen mentioned, I've had the great fortune to be able to have amazing students, a lot of them through the Honors College, not only work with me, but work with many members of the Linus Pauling Institute, and they are certainly a big part of our success in our research, and more importantly, the impact that the center and the Institute have on health. Kevin, are you able to share my slides? Perfect. So I want to talk to you a little bit about the Institute. So here's our building. We are celebrating our 25th year at Oregon State University, and the Institute really tries to hold up the name of Dr. Linus Pauling. Do you want to advance to the next slide? Our mission is to promote optimal health through cutting-edge research and trusted public outreach. And we really like to talk about helping discover, but also enable populations to be able to reach their optimal health. We often talk about how can we help people take back their health, largely through food, nutrition, and other things that they have control over. Some of our priority areas I'll talk about in a little bit more detail are healthy aging, precision health, data sciences, and then the last piece is health literacy and outreach. We feel strongly that our science needs to go past our lab bench, and it doesn't. We need to make that impact by ensuring the information that we have and the science that we discover is accessible to the population, not only in knowledge but also in action. Next slide. The other big thing that I want to emphasize, so similar to the Honors College, we're able to draw upon the expertise of multiple disciplines and really leverage the multifaceted expertise across Oregon State University to really understand the mechanistic determinants of optimal health. So our faculty and our students come from all over Oregon State, pharmacy, veterinary medicine, public health, College of Ag, engineering, all together, working together to solve some of the biggest problems that we have, especially in health. Next slide. I mentioned healthy aging is one of our priorities. And when I talk about healthy aging at the Institute, we're talking more than just living longer. It's not just longevity. We're talking about living longer, but more than that, we're talking about living better. So we have this concept of health span where we really want to optimize living your best life for the longest period of time. And this is something that Linus Pauling also really endeavored. So, how do we optimize your mental capacity? How do we maintain a healthy immune system? How do we stop disease? And if you're diagnosed with one of these diseases, how do we slow the progression so you can live with the disease, not necessarily die because of it? And how do we best manage it? So again, it's really about living better longer. Next slide. For those of you who might not know a lot about Linus Pauling, he is one of the only individuals to hold more than one unshared Nobel Prizes. And I want to emphasize that one of his prizes is in science for the nature of the chemical bond, but his other prize is a Nobel Peace Prize. And we really take that inspiration in terms of what we do. We want to discover the science, but we also want to make sure that we have that impact. So another big piece that we have students also involved in is that getting the science message out to the public as well. Next slide. So we have a resource called the Micronutrient Information Center that has been our flagship outreach program that helps really communicate our science and is an evidence base in terms of understanding micronutrients and their role in disease processes. And this is another big piece of that full circle. We're very committed to training the next generation in terms of our undergrads and research, but we're also committed to making sure that our students are able to amplify their message.
Emily, your mute keeps going on and off, so.
Sorry about that. I'm not sure what you heard, but the main thing is that we want to make sure that our students not only discover, but become champions for their science and are able to amplify their science by making this impact and propagating what they find to the general public. Next slide. So the bottom line is, this is again a quote from Linus Pauling. We're really here to help you live better longer. We provide the science, we provide the education, and again, we've been so fortunate to be able to have partners like the Honors College to help us reach our goals and reach as many people as we can in terms of our message. So we've just loved having the Honors College students with us. And again, it's a true partnership where those students are really our colleagues and partners and champions in health.
Thanks so much, Emily. There's a quick question in the chat that, if you don't mind, I'd like to ask you and see if you can respond to it. I think it was Dean Moore asked, what's precision health? Do you mind responding to that?
Yeah, so precision health kind of talks about, so when we think about dietary recommendations, for example, there are not blanket recommendations that are meant to be kind of one size fits all recommendations. A lot of these recommendations are based more on our essential health, how to prevent deficiencies, vitamin C, prevent scurvy. What we're really looking for is that optimal health, which also means that those recommendations may be slightly different depending on your situation. It could be based on your genetics, your family history. It may be based on where you live. So we are really looking to see how we can incorporate all of those factors that contribute to your life to be able to better precisely make recommendations that fit for you. And that may change over your lifespan as well.
Wonderful. Thank you so much. And so now it is my great pleasure to welcome our 8 student presenters. As important as research is for a student's own development and education, these presentations also make clear the quality of the research and creative work that's being done by honors students. They're making real contributions to their fields. These contributions have significant impacts and implications. Our presenters are all at varying stages in their research process, but together they truly do showcase the diversity and the depth of research and the accomplishments of researchers at Oregon State University, as well as researchers in the Honors College. We've asked each of our students to speak very briefly for just around 3 to 5 minutes. And so this is part of the reason we don't have faculty do this, because most faculty don't know how to speak for only 3 to 5 minutes, but our students do an amazing job. And what we're going to do next is we're going to go through all 8 talks without breaking for questions. And then after all 8 students have finished, we'll split into 4 different breakout rooms. And we've assigned 2 students in each of the breakout rooms. And then each of you has been assigned to one of the breakout rooms as a starting room. But after you ask questions of students in that room, you'll be free to leave and visit the other rooms. If you have any difficulties, Trisha Riley is going to stay in the main room to help make sure people get into the breakout rooms that they want. So you'll have a little bit of time available to join with different students and ask questions of any students based on the information that they are going to be sharing with us. And we're also going to post a link with a website to all of the slides and titles and a list of which speakers are in each room. So we're going to go ahead and get started. And so now it is my pleasure to invite AnneMarie Hasbrook to begin our presentations.
Hello, thank you. Hello, my name is AnneMarie, and today I will be presenting on thrombogenic comparison of commercial and bio-inspired SLPS coatings. Next slide, please. A universal coating with anticoagulant effects is needed for a photoreactor device. This device will run blood under blue light to help break down bilirubin buildup. In order to maintain proper blood flow throughout the device's tubing, a coating that has antithrombogenic properties to prevent blood clots is required. This eliminates the need for oral or injected anticoagulants. Next slide, please. One method that can be used to create this needed coating that contains the anticoagulant properties is a bio-inspired SLPS coating. The bio-inspired SLPS coating consists of a layer of polydopamine, which is inspired by the adhesive properties muscles possess. The DOPA and lysine histidine adhesion and mobilization layer allow for the next layer of grafted functional thiol layers to attach. The outermost layers consists of a SLPS oil, which is inspired by the oil slippery pitcher plants produce to repel both solids and liquids. This oil provides a slick, repellent, robust, and self-healing surface when in contact with substances. Next slide, please. To assess the antithrombogenic properties of this bio-inspired SLPS coating, an in vitro model is used, specifically the Chandler loop. The Chandler loop provides a means to mimic blood vessels and flow outside of the body by rotating the blood at a desired flow rate under physiological conditions. The model compares the SLPS coating, uncoated Tygon tubing, Livanova cardioplegia tubing, and Teramos cardiovascular procedure kit tubing. Next slide, please. And could you press next again? Thank you. So here's a short video showing the Chandler loop model in action. As you can see, the blood flows opposite of the rotational motion, and the connection points are sealed using latex tubing of a fitted diameter. Next slide, please. Different parameters being tested to assess the coating's compatibility with blood are when the first clot forms and where, when blood flow stops, and how much of the surface area the clot occupies. Next slide, please. The first set of experiments consisted of timing the duration of flow within each tubing until the first clot appeared and when blood flow became obstructed. The graph shows a comparison of the 4 coatings being tested, with the Tygon tubing forming a clot and obstructing flow first, and the Livanova cardioplegia kit being the last to form a clot and obstruct. The slips coating clotted and obstructed between the Tygon and commercial tubings. Next slide, please. After flow and the tubing obstructed, they were removed from the Chandler loop and cut into 1-inch segments to analyze the surface area of the clots compared to the total surface area within the tubing. The clot coverage was measured using image J to calculate the area. Next slide. Using the clot area and total surface area data from image J, a clot coverage percentage at each position was calculated and graphed to analyze clot formation. The connection points of the tubes are located at positions 1 and 16, and the data shows that the highest clot coverage occurs around the connection points. It is important to note that we are still in the preliminary stages of the experiment, and only one trial has been run. Despite the lack of repeatability, some conclusions can be drawn. The Livanova commercial tubing took the longest to form a clot when on the Chandler loop, and the slips coating had the shortest time interval in between the appearance of a first clot and flow obstruction. This indicates that the Livanova commercial tubing withheld antithrombogenic properties for the longest duration. The slips coating also showed the highest clot to surface and coverage, likely indicating low antithrombogenic properties, but still performing better than bare Tygon tubing. A common trend throughout the coatings was the position of the first clot formed and where the most clot coverage occurred, which was near the connection points on the loops. This could indicate a need for a better method to seal the tubes. Overall, more work is needed to better understand the coating to blood interaction. Moving forward, I plan to repeat the experiments more times in order to gather more information about the various coatings' hemocompatibility. I plan to analyze various methods to connect the tubings while on the Chandler loop, as clots began to form first at the connection point, causing the clotting cascade. And eventually, I plan to investigate various blood clotting components such as thrombin concentration in order to better understand the interaction between the blood and the different coatings. Thank you all for your time, and I would also like to formally thank members of my lab group, Madeline Hummel, Dr. Ryan Faze, and Dr. Andrew Carpenter for their help throughout the process, my thesis mentor, Dr. Bayo, and the Honors College for inviting me to present today.
Thank you so much, AnneMarie. Next up is Anna Brousseau.
Hello, my name is Anna Brousseau, and for my Honors College thesis, I'm studying CRISPR-Cas9 genetic engineering in poplar trees. So in order to genetically modify plants, a natural bacteria known as agrobacterium is used to shuttle the CRISPR machinery through the cell wall and into the nucleus of the plant. The integration of transferred DNA into the genome then creates a transgenic plant. However, it's desirable to be able to make edits and then remove the transgenes. So 2 reasons to make a transgene-free plant are to comply with biotechnology regulations and avoid further undesired mutations. The method of transgene removal being tested uses the enzyme Cre recombinase, which can remove large pieces of DNA and hopefully the integrated transgene. The recombinase activity can be controlled by placing a promoter in front of the coding sequence for Cre. Hoping to find an ideal promoter, I studied the 2 Arabidopsis promoters of Wuschel and cold shock protein 3, which are naturally found in primordial cells. Instead of controlling Cre recombinase, Both promoters were then placed before the coding sequence of green fluorescent protein, which allows the promoter's activity to be tracked and determine how well the promoters could control Cre. The timing of Cre activity is extremely important because it's necessary that the transgenes have enough time to make the genomic edits, yet need to be eliminated before there are too many transgenic cells. So here are the 6 different stages of tissue type that were observed. First, on the left is callus tissue, which was developed and is an undifferentiated stage that helps change the explant from stem to the subsequent 5 different shoot stages. So the ideal model is displayed here. The yellow line represents the presence of transgenes in the plant, and the green line shows the amount of Cre activity being controlled by the developmental promoter. So as you can see, as the activity of Cre increases, the transgene should then be removed. So in this experiment, cuttings of young poplar stems were placed on 2 growth mediums to induce development. Weekly, I examined 200 explants under a fluorescent microscope and recorded which tissues were visible. The GFP brightness of each tissue was also recorded using this scale, ranging from 0 on the left being no GFP signal to a bright 3 on the right. So by editing the genome, we can change specifically targeted genes, and each random insertion creates what's called a gene insertion event. So here you can see 3 different events. And after tracking the GFP signal and averaging the brightness over all of the events, it was found that AT-Wishel promoter had really high callus expression and then a decrease in brightness throughout the rest of development. Comparing this to AT-CSP3, which had a lower calli brightness and then a significant increase during the early shoot stages of development, before then decreasing over later shoot stages. There was also very high variation between events of the same promoter, shown by the high coefficient of variation and exhibiting that neither promoter was consistent. So comparing both promoters to the ideal model, you can see that neither fits perfectly. So these findings further display that the process of regeneration is messy, and finding a promoter that exactly matches the ideal outcome is an ongoing challenge. To successfully use either of these promoters in the future, they would have to be combined with other techniques, or alternative excision methods would need to be pursued. Finally, I would like to thank everyone in the lab who has helped me and continuing to help me with this project. Thank you all for listening.
Thank you so much, Anna. And I expect all of you, like myself, are already starting to come up with some interesting questions to ask our students. Next, we have Annelise Hartinger.
Hello. So my research, we're studying the effects of COVID-19 on individuals with rare disorders. And my mentor is Dr. Bogart from the School of Psychology. So what is a rare disorder? So a rare disorder is any disease or disorder that affects less than 200,000 people per year. And while there's not a lot of people who are affected by each specific rare disorder, there are more than 7,000 types of rare disorders. And these are just identified rare disorders. So when you combine all of the rare disorders together, approximately 1 in 10 people in the US do have some kind of rare disorder. So for this study, we were building on previous research that shows that people with a rare disease or disorder already suffer from a lack of understanding, lack of accessibility, lack of activism, and a lot of other challenges. And we want to know how COVID specifically added barriers or maybe potentially limited barriers for this population. So we collected open-ended responses from 759 participants who were all adults with some form of rare disability or disorder from May to July of 2020, so right when the pandemic was first starting. Each participant was asked 9 open-ended questions, and with these questions, we were trying to gauge impact and difficulties, healthcare access, coping, silver linings, and cultural or societal changes they've seen happening or think will happen as a result of the pandemic. They were also asked some demographic information, but we mainly focused on the open-ended questions. And our study is a qualitative, inductive, data-driven thematic analysis, which means we start with the qualitative data, which is the response for each question, and then we code it into these groups. So we group them together based off shared ideas or themes that we're noticing from the responses. And then we take those codes and we group them again into major themes. And then we use that as our big comparison to show the major themes coming out of this data. Something important to know with qualitative analysis is each quote can have several codes or no codes. So just because we had 759 participants, they had up to 9 opportunities to respond to questions. That doesn't mean that each had 9 codes. They could have had several codes for each question or no codes. So it creates quite the variety of our results. So in our study, we end up with 3 main themes: systemic and health factors, coping, and support. So systemic and health factors, we're looking at anything that affected mainly the healthcare system, but any society-wide systems. Coping was how the individual is coping with the pandemic, and support is how they are receiving support from others or the community during the pandemic. We also had a code that does not belong in any of the themes called no noteworthy issues. And this is where they had no issues with that related question, or their experiences weren't different from that of people who don't have a disability. That does not belong in one of our themes because it's not addressing the question. Just want to note that here. So systemic and health factors was our largest theme by quite a bit. The upper quote is in regards to public health, and the lower quote is in regards to negative healthcare effect, if you want to look at that. So each of the numbers next to these codes represents how many codes we had for that. So that's how many responses met that code. Healthcare negative effect was not only our largest code in this theme, but also our largest code in our entire data set. So this really indicates that the biggest effect we've seen for people with rare disabilities during COVID is a negative impact to their access to healthcare. And so this is including things like limited access to medication, limited access to doctors, treatments being stopped, shortage in medical supplies. So anything like that, anything that's negatively impacting their ability to perceive healthcare or take care of their own health is in that code. Our next theme is coping. The top quote is in regards to resilience, and the bottom quote is in regards to distress. Distress was also one of our bigger codes, and that is simply any negative emotion expressed: anger, frustration, anxiety, fear. Any of that would be in distress. One of the interesting patterns we saw with distress was that people felt that the pandemic showed how society valued them. That with this pandemic shows that people with any disorder, but specifically a rare disorder, are viewed as less valuable members of society than able-bodied persons. So this is one of our interesting patterns we've noticed with our data. And our last theme was support. Family was our second largest code of all of the codes we had. The first quote is in regards to informational support, and the bottom quote is in regards to family. So family was anything in regards to your family, spending more time with them, spending less time with them, driving nuts by your family, getting support from your family. Anything with your family is family. Interesting thing to connect to is companionship and support is also one of our big codes. And that was basically the same kind of code, just not by your family. So that's friends, support groups, partners, pets. So those were very similar, just kind of which area that support is coming from. So it really shows how people with rare disabilities during this pandemic really derive support from those they have immediate connections with. That was the biggest source of support for our population during this pandemic. So what did we learn from our study? We're still in the process of writing up, but we have finished collecting and coding all the data. So the largest impact we saw was to healthcare. And while it was mostly negative impact, there were some positives. The biggest impacts was medical care impediments, reduced availability of providers, medication shortages, insurance issues, and an increase of symptoms. Usually the increase of symptoms derived from either the stress of the pandemic or just from the lack of access. So it kind of was a confounding factor. But there were some positives. So people did experience greater support from friends and family. They believe that they are feeling greater inclusion and or understanding, mainly from their immediate support group, as they do. There is some society-wide issues that we're also seeing. There's greater access and accommodations to both work and the public. The big one was working from home. That did benefit a lot of people with rare disabilities to have that option available to them. And they're hoping that the pandemic allows improved public health measures moving forward to aid the rare disease community. And we're hoping that this research, once we're published and get it out there, will help improve public health measures, help inform government policies and or benefits, and help make advancements in the medical field. We do have a lot of quotes and participants asking for advancements, not only in like medicine and treatment options, but in how they are working with their healthcare teams. Some exciting news of the study is we were just accepted to present at the Society of Behavioral Medicine in April. So we'll be able to share our research on that platform as well soon. Thank you all for listening to the presentation.
Thank you so much, Annelise. Maya Livni is our next presenter.
All right. Hello, my name is Maya Livni, and today I'll be talking about the impact of selected gut metabolites on anxiety behaviors in an autistic mouse model. Our gut is home to about 100 trillion bacteria. They digest food, they regulate our hormones, produce key nutrients, and play a large role in the body's immune defense. An abnormal gut microbiome has been associated with several central nervous system disorders, such as multiple sclerosis, Parkinson's, and autism spectrum disorders through the gut-brain axis. The gut-brain axis is the two-way communication system that exists between the central and enteric nervous system, and which is influenced significantly by microbes in the gastrointestinal tract and vice versa. However, despite the key role that gut microbiome plays, relatively few gut metabolites have been identified, and minimal treatment options exist to remedy an unhealthy gut. The project I'm presenting today is a collaboration with the company Second Genome and Stanford University. In this study, several gut metabolites were found to be depleted in the gut microbiome of individuals with autism spectrum disorder when compared to their neurotypical siblings. These metabolites were then fed to mice with an autistic phenotype in order to investigate potential therapeutics. We fed our mouse model, catnap mice, with a diet supplementing different metabolites. We conducted this feeding regimen each night for 2 weeks leading up to behavior testing, as well as through the approximately 10 days of behavior tests. We conducted 4 different behavior tests, including elevated plus maze, 3-chamber sociability, marble burning, and habituation tests. We performed these on cohorts of 12 male mice each to determine if each specific compound had the capability to decrease behavioral states, such as anxiety, when compared to the control mice who were fed mice chow without the metabolite. The analysis of behavior data was done with the help of Behavior Cloud software and RStudio. One of the behavior tests we performed was called the 3-chamber sociability and social novelty test. Neurotypical mice prefer to spend time with another rodent, and they'll investigate a rodent intruder. This test uses this premise to test for deficits in sociability and social novelty in mice with an autistic phenotype. This test is broken down into 3 10-minute stages. In the first stage, the test subject is placed in a 3-chambered box with openings to each chamber and is allowed to habituate or explore the apparatus. The second stage tests sociability, identifying if the mouse prefers an empty chamber to the chamber with another mouse in it. The final stage tests social novelty, examining if the test subject prefers the first or the second intruder mouse. For example, the 3-chambered sociability test revealed that mice fed with compound A spend significantly more time with the more novel intruder in the 3rd stage of the test. This implies that compound A may have the ability to help bring an autistic phenotype nearer to a neurotypical mouse phenotype. During each of our 4 behavior tests, we collected similar data, such as measuring time spent and distance traveled in different regions of each maze. We, in addition, measured the number of entries into different areas of different sections of the maze. Currently, behavior testing is complete, but in the future, we will examine possible mechanisms of action for the various compounds we studied by using the tissue samples we collected at the end of behavior testing and experimentation, and we'll conduct 16S amplicon sequencing on these samples to determine the microbial community of the gastrointestinal tract. I'd like to thank the following members of the David Lab, especially the graduate student Alex Phillips, for helping and supporting me in my research endeavors.
Thank you so much, Maya. Now, Shaurya Gaur will present.
Hi, everybody. I'm Shaurya Gaur, and I'm studying how to use machine learning to create more dynamic and productive music playlists. Listening to your favorite music makes you feel a certain way, whether it's slow and sad or upbeat and excited. That's why services like Apple Music and Spotify make dozens of playlists based on our moods. Music also has an effect on our focus and productivity, but most playlists aren't designed to lead users towards that. Prior studies on emotion and music often sought to create playlists that keep users at a specific mood rather than moving between them. We seek to create a way to make playlists that take users from their current mood to a more happy and focused one. That way, playlists meet users where they are and gradually move them to a more productive state. To chart this journey, we based our research on the arousal valence circumplex model, a popular framework for classifying emotions by their intensity, also known as the arousal, and positivity or valence. Its point-based approach will work well since our playlist is basically considered a path of songs in this space. We originally tried the DEEM 2016 dataset consisting of 2,000 free public domain songs represented as coordinates in the arousal valence model. However, we later switched to the Deezer 2018 dataset consisting of 18,000 songs covering 2,700 unique points in the arousal valence model with arousal and valence scores calculated based on the song's last FM tags. We believe that the greater number of songs, their availability on major streaming platforms, and the more varied range of points will make our playlists more applicable, larger, and more varied. So our goal with our model is to start at the user's current emotional point and create a path to a destination point in the arousal valence space. This emotional path should be smooth, and songs in between should be evenly spaced. This all ensures that no one song is too drastic a change from the previous and that the tone changes gradually. We're using a k-nearest neighbors machine learning model for our algorithm since it's designed to handle point values. To choose our songs in the playlist, we first find a hypothetical target point in between our current and destination points. We then find the k points nearest to our target, calculate the distances to those points, and then use a distance metric to determine which point is most similar to our target. That becomes our next point, and we keep doing that until we reach our destination. We tested several distance metrics to make sure our playlist was as smooth as possible. We compared playlists of different lengths to a perfect line between the origin and destination points using the popular mean squared error metric. From our tests, we found that using cosine similarity generated the smallest error and therefore was the smoothest. You can see when on a side-by-side comparison to the popular Euclidean distance on the right, the playlists that we generated with cosine similarity were generally much smoother. And I'm going to change my screen share really quick so that way we can take a look at an example. So here, let's take a look at our physical, like our playlist itself. Do you guys see a Google Chrome window? Okay, I see some nods. So here on this website, we can choose our starting and ending songs, as well as the number of songs we want on our playlist. You can find the link for the website in the chat, which I'll put after the presentation. So we can set our origin song to something really sad. So one that I always like to do is this one, How Can I Help You Say Goodbye by Patty Loveless. You can see that the arousal and valence points are pretty negative. So this song is not only low energy, but also pretty sad. And then we want to go to a high energy, high emotional song. So let's go to 1963 by Rachel Yamagata. And we want 15 songs in our playlist. So we can create our playlist and we get a list of songs right here. And we can find the playlist on Spotify if we click this link. And there we go. And let's switch back to the presentation at this point. There it is. So we can track, so we click submit, the algorithm took that data in and it created a playlist. And now we have the list of songs. We can see the list on Spotify, and we can also, I've decided to plot all of these in the slides as well to walk you through it. So first, the songs start off as relatively sad, and we see that they slowly creep up to become more positive and higher energy. And so they'll keep going until they're a bit more neutral emotionally and have a neutral energy. And then they'll eventually get to a point where they're not only very positive, but also have a high energy. As we can see, our algorithm successfully generates a smooth, direct path between two emotions in the arousal valence space using music. This work is a starting point for further research. Currently, we are also working to integrate our playlist with neural networks that use facial emotion recognition to identify a current user's emotion. And we're also using Spotify's built-in audio features for songs, which they use to classify genres, to create playlists that not only create an emotional journey, but also stay within, say, your music taste. Playlists like these can be powerful tools in mental health and productivity, gently guiding users to happier or more focused states of mind. At the end, I'd like to thank all of you for listening, and I'd like to thank my lab, especially my mentor, Patrick Donnelly, and who also recently worked with me to submit a paper to a conference that we'll be going to in April. Thank you very much.
Wonderful. Thank you so much, Shaurya. Next, we have Nikita Rubocki, who is one of our Honors College students at the Cascades campus. Nikita.
I will share my screen real quick. There we go. Perfect. Can everyone see that? Sweet. Hello, everyone. My name is Nikita Rubocki, and my thesis is called Dealing with Data Sets. It's actually based off a project I created at work called Feature Analysis, and it's within the scope of the data science machine learning industry. Oh, let's see, there we go. Next slide. So within this industry, there's a funny quote that says that 90% of a data scientist's work is in data preparation. In other words, many data scientists spend most of their time trying to understand a data set. For those of you who are unfamiliar with the data scientist profession, the actual main job of many data scientists is to train a machine learning or artificial intelligence model to predict on the outcomes in a data set. So if they're spending most of their time cleaning data or preparing data, then only a small fraction of their work goes towards their actual job. Furthermore, once they have data they can work with, they spend a lot of time trying to find the perfect model for that data. Depending on the task, data scientists may run up to hundreds of thousands of models looking for the best one. And depending on the model, this can take a lot of time. In short, a large portion of data science work goes towards data preparation and then running models on that data. What if both of those tasks could be completed more quickly so time could be spent on more important things? Feature analysis is an innovative new way of addressing this pain point. What exactly is so hard about analyzing data sets in the first place? Let's start with a simple example. Here you can see a very basic data set I created which holds Apple data. The column we are trying to predict, in this case the type of apple, is called the target or class label. This is basically the correct answer to a row of data, known as an observation, which a machine learning model will try and will learn and try to ultimately predict. All the other columns are known as features of the data set. This data set has 4 features total, some of which are more clearly correlated to the target label than others. For instance, the ID column tells us nothing about the apple type and can likely be discarded from the data set before being fed to the machine learning model. On the other hand, the height feature looks like it might be a great predictor of apple types. Seems simple enough. However, not all data sets are this clean and easy. Many real-life data sets start off dirty, meaning there might be data missing, features may be inaccurate or incomplete, and the data itself may need to be standardized or normalized to prevent bad or biased models. Additionally, it's usually difficult to understand the correlations between features. That's why we invented machine learning in the first place. As you can see, understanding and analyzing data sets is very important. However, data is only the first half of the equation. Once a data set is cleaned, it's time to create the best model for it. This slide here shows the basic steps to finding an optimal model for your data set. What does it mean to find an optimal model? Every model has adjustable parameters, known as hyperparameters, that can be adjusted to change how the model behaves. A data scientist may run many different models with varying hyperparameter configurations to find an optimal model, or one that will predict well in the data set. Again, though the number of models run depends on the problem, I've personally run hundreds of thousands of models to find an optimal solution for one single data set, which amounts to about 200 hours of runtime. Yes, that's a lot. Given all of that time and effort, I thought there must be a better way. So I developed feature analysis. Though the mechanics can be complicated, the basic idea is simple. Use machine learning to find out which features in a data set are the most important or most highly correlated to the target label. After taking a data set and running it through a collection or ensemble of machine learning models that include a function called feature importance, I run the results through a specialized ranking system, which results in the output in the final box. So in this simple example, assume the data set has 5 features, A through E. The final box tells us that feature C seems to be the most important one. You can see it scored the highest in relation to everything else. Feature E, on the other hand, scored the lowest and can likely be discarded from the data set without affecting any results coming out of the model. But wait, how are you training those ensemble models if finding optimal hyperparameters is the main point of feature analysis? It sounds like a bit of a chicken and egg kind of scenario. This is where my thesis research comes into play. Based on preliminary results, the feature analysis tool can be used to reduce features in a data set, increase interpretability of results, and provide better insight into model hyperparameters. All of these lead to improved dataset understanding and finding an optimized model faster, which are the 2 big problems I mentioned previously. However, this program is in the early stages of research. Though it's been used on a handful of datasets and models, the scope and overall methodology of the program still needs to be tested and verified with different datasets. My thesis is focused on this task, working to answer one basic question: does feature analysis truly work? This is no small feat, as data scientists are rigorous and data-driven people, and they expect their tools to be as well. Through my thesis, I hope to verify that feature analysis can be used as part of a data scientist toolkit, providing better insights for the data science industry at large. This way, datasets become easier to wrangle, leaving more time for the groundbreaking work that truly matters. Thank you all for listening to my presentation today.
Thank you so much, Nikita. Next, we have Darlene Nguyen.
Hi, everyone. Let me just present this really quick. My name is Darlene Nguyen, and this last summer, I worked with Dr. Najee Bond on our Ethnic Studies National Survey and K-12 Institute project. This summer, I wrote scripts and recorded videos that define ethnic studies terms and concepts. The video essays that I scripted and recorded were male and white gaze theories, minstrelsy, code switching, and internalized oppression. For the latter part of my project, I used survey data collected by the U.S. Department of Education to create digestible charts and graphs. This process helped me understand where ethnic studies majors and resources are concentrated in the country. I also got to interview James Jeffrey West from the Portland Public School District to get a sense of how ethnic studies has been integrated at the high school level. The survey data revealed to me that Oregon held 12% of all ethnic studies degrees in the country in 2018, but the reality is that that is only 21 degrees. And this part of the project really gave me a reality check on how little ethnic studies has been integrated throughout the United States. And I wanted to look, I wanted to look deeper into how Oregon may be fostering an environment where students want to pursue further education in ethnic studies. I discussed the pros and cons of making the majority of these classes electives with Mr. Jeffrey West. And while students may be more enthusiastic to take these elective classes because they're lower stakes, many students may still cycle through the school system without this perspective because it's not quite embedded into the core curriculum just yet. And while there are limitations, this is actually extremely progressive relative to the rest of the country. For many states, ethnic studies materials and resources are only available at the college level, if at all. I encourage educators to look into how they can share knowledge that they've obtained in a way that's accessible to all. And that's the vision I had going into creating these videos. And I wanted to share a short clip of what I worked on. Centering whiteness is only one aspect of the white gaze. It could also look like stereotypical depictions of non-white characters.
Take Cho Chang from the Harry Potter series, for example. Chang is the only East Asian character in the series. She's a quiet, school-driven Chinese girl. The main purpose of this character is to serve as the token Asian wizard and the submissive love interest of her white counterparts. Mistakes, such as naming her 2 Korean surnames, go undetected under the white gaze because the names Cho and Chang are immediately perceived as Asian, which is all this character was meant to be seen as, an Asian girl in the background of white protagonists. So, while representation matters, it's only half the battle. The white gaze reveals to us that even when there is an inclusion of people of color in film, their role may be tarnished by a racialized bias. Understanding the white gaze and different gaze theories allows us to see beyond the author's subjectivity. The constructs.
And as I continue to read and learn more about ethnic studies concepts, I can see why students whose grasp of racism is that it died a long time ago with segregation have an incredibly difficult time seeing how race and racism affect us right now. And it's so easy to dismiss them as just bad apples. We must ask the question of what larger systems are fostering these ideas. It's because our public education has not integrated anti-racist work into our curriculum enough. A lot of U.S. history curriculums exclude perspectives and stories of people of color. And while I was writing these scripts to these videos, I was constantly asking myself, how can I make others see beyond this whitewashed history? And using references that are reflective of student experiences and pop culture allows for people to recognize biases even in the ordinary. And these videos can be used by school teachers who want to explore these concepts or curious onlookers because these are posted on a public platform. And I have hopes that the pop culture references are more likely to make it outside of the classroom and into conversations with friends or at the dinner table with family. Ultimately, I believe that we must embrace necessary changes to our country as they're being voiced by our most marginalized communities. Our education system has been so dramatically impacted by unquestioned patriotism and colorblindness. And I believe that integrating an ethnic studies lens into public education is the first step in doing so. This is the vision that I had, but we cannot get there without providing people with the tools of education first. And I see these videos as exactly that. A first step. And I'm grateful to be given the opportunity to create these tools and share them on a large platform. Thank you so much for your time.
Thank you so much, Darlene. And now for our final presentation, we have Sophia Fischer.
Okay, so my name is Sophia Fischer, and this past summer I worked on the project OSU's Latina/Latino/Latinx Social Justice in Oregon with my mentor, Dr. Zapata. The purpose of this project was to interview Latinx social justice leaders and allow them to share their struggles with marginalization and their impact in the state of Oregon. This project is ongoing as Dr. Zapata and his students are still conducting interviews. The overall goal is to launch an online database with digital video and audio interview clips that would be publicly accessible. For this project, I found interviewees, many of which were from different nonprofit organizations such as Bakun, Capaces Leadership Institute, and Latino Network. I interviewed 16 people, wrote biographies and synopses, and categorized key terms. The oral histories from this project have specifically allowed interviewees to share the struggles of marginalization, but overall, oral histories are significant because they allow everyday members of the community to share their voices, which can be often a scarce opportunity. In society, we like to generalize people into different groups, but these oral histories emphasize each person's individual story and their powerful life experiences. And the way I wanted to share my project was through some quick quotes to preview these interviews. I'm not Latinx, so I don't have the experience of Latinos living in Oregon, so I cannot comprehend what they have faced. But these individual oral histories exemplify these experiences. I wanted to start with Ana Munoz and a quote of hers about seeing the Latinx community through its individual members.
A lot of times, when it comes to social justice, we don't think about our people as individuals. We think about people as percentages and categories and really labeling them. And when it comes to addressing people as individuals, you get a whole different perspective. And
she goes on to describe how the Latinx community is often used and abused, and they never hear how their information is used in the media. The state of Oregon is currently making efforts towards diversity, but simply getting one spokesperson for an entire community is not enough. Since the purpose of his project was about social justice, many interviewees also shared their own personal experiences with racism. In this clip, Inés Peña describes her experiences living in Newburgh throughout her life.
There's this niceness to folks that I would say was the norm, which things changed, as the only constant is change. And things really changed after the Trump administration, because that nice politeness that I experienced growing up was no longer there. Some of the people that were nice and polite flipped the switch and really were racist. And I think the Trump administration gave that blanket permission to be open about the racism.
This clip explains how, although there have been recent changes, working towards social justice is difficult because society at times does move backwards as well. Some of the other struggles that occurred in the interviews include people describing their experiences being threatened, applying to college without guidance, living with the fear of deportation, difficulties of being a dreamer, and facing discrimination in the workplace. Something else I wanted to exemplify was the cost of moving to the United States.
I think that many people come to the United States because of the opportunity and because of freedom, but that freedom and that opportunity comes at the expense of other people, and sometimes we disconnect those things. So.
People often have this dream of freedom and opportunity, the American dream, but they don't recognize that it comes at a cost. And as Joanne Mina describes, it comes at the expense of other people. There were many goals mentioned in these interviews, but I specifically wanted to highlight the farm worker community because of the injustices they face.
That we do there, and I think that for too long, we, as a state, as a country, have turned our heads and backs to these injustices and unlawful practices. So I want to see us move forward, and that comes to the humanizing and seeing Black and Brown people, regardless of the job that they may hold, as humans who deserve dignity, who deserve respect, who deserve a safe work environment and a safe place to live and safe community.
These farm workers still do not have collective bargaining rights, the right to unionize, and are unable to fight back against their superiors that go against the law. There are many more powerful moments in these interviews, and I hope you're all able to watch all 16 of them when they're released on SCARC and the website when the project is finished. Thank you.
Thank you so much, Sophia. Wow. I know that all of you probably have more questions than we have time for. So really shortly here, we're going to be shifting into our breakout rooms. Since we're a little bit after 6 o'clock, we'll keep those open for a little bit longer than we had originally planned. We're going to put in the chat a guide to which rooms that the different students will be in. And Trisha Riley will remain here in the main room to assist you if you have any issues getting into the room that you want. And then I want to thank everybody for joining us. A special thanks to Dr. Ho for your remarks and helping us all learn a little bit more about the Linus Pauling Institute. And a special thanks and a shout out to our student presenters. We are in week 7 of the term, so I know that they're all really busy, so it's great that they made some time to be with us tonight. Many of our students have benefited from scholarship support, and we're truly grateful to the donors who help make the honors experience possible for our students. If you'd like to learn more about how to support student research in the the Honors College, you can reach out directly to myself or to Kelly Parker at the OSU Foundation, and we'll try to put that information in the chat as well. And so you all should be moved automatically, momentarily into a breakout room. And again, once you're in a breakout room, you'll have the ability to switch to any of the other rooms. Thank you so much.